So today the basic process that we are going to look at is trans-deamination. We looked at transamination. Trans-deamination includes transamination followed by oxidative deamination. Now we would look at the overview. First, all cells which are present in our body convert one amino acid into another. Mostly the second amino acid is glutamate. Now it has to reach the liver, as liver only has glutamate dehydrogenase enzyme. So glutamate or glutamic acid is converted to glutamine. When it reaches the liver, it is again converted back to glutamate, undergoes deamination to give ammonia. Ammonia is toxic, enters urea cycle and excreted out. So this was overview which we had seen previous lecture, but I showed it again over here so that it is remembered much better. Now we'll look at deamination process. It has two types, oxidative deamination and non-oxidative deamination. Non-oxidative, which is there, it is shown by those groups which do not show transamination. Which were those groups which didn't undergo transamination? First were, in transamination, not everybody gave the ammonia, gave the glutamate group, right? Now we need ammonia from the amino acid, so anyhow we are going to use this process to get that ammonia. That is serine is one of it, which undergoes the process with the help of dehydratase to give the ammonia. Again there is threonine, which is also under the pressure of the enzyme dehydratase gives ammonia. So these two, that is OH containing group, these two are the examples. The third example is of sulfur containing group, that is cysteine. Cysteine, which is there, it is also going to give ammonia in the presence of the enzyme desulfhydratase. Okay, the enzymes are different, but only thing to remember here are the examples. And the third and the last example which is going to undergo this process is histidine. Histidine in the presence of histidase gives ammonia. So the four examples which we are going to remember here are OH containing, sulfur containing and histidine. These undergo non-oxidative deamination because they do not undergo transamination, while rest all underwent the transamination and are going oxidative deamination. Now the thing that we need to study over here is oxidative deamination, and oxidative deamination is the process which takes place where the glutamate which we had received is going to be broken down. Glutamate was the amino acid that we had received, right, from the transamination process. All the amino acid are first transaminated to glutamate, and this glutamate that we have received is going to go to the liver because the enzyme glutamate dehydrogenase, which is breaking down glutamate, is present only in the liver. So when glutamate breaks down in the liver, it first gives a compound, an intermediate compound, which is not much important. Here this glutamate dehydrogenase converts NAD plus to NADH plus H plus. The enzyme which is used is quite important. It's present only in the liver. I have emphasized it a lot of times because it's important. The intermediate is alpha aminoglutamate, which again undergoes the reaction in the presence of GDH and water to give alpha ketoglutarate and ammonia. So ammonia is important, which goes or enters the urea cycle. So in this process we got ammonia. So this process which is there occurs in the mitochondria of the liver. The entire process occurs in the mitochondria of the liver. Now glutamate dehydrogenase enzyme performs a reversible process. If it goes ahead, it can come backwards as well. And how is the regulation process of GDH taking place? First of all, here allosteric regulation takes place. It is activated by GDP and ADP, and it is inactivated or inhibited. Its action is inhibited by GTP and ATP. So it's simple, activated and inactivated. Activated GDP ADP, inactivated is GTP ATP. Now after regulation with the help of this enzyme, we look at two oxidase which are present in our body, L-amino acid oxidases and D-amino acid oxidases. In L-amino acid oxidases, the coenzyme which is present is FMN, flavin mononucleotide, and for D-amino acid oxidases it is FAD, that is flavin adenine dinucleotide. FMN is the coenzyme. Okay, the enzyme acts on all the amino acid. L-amino acid acts on all amino acid except for hydroxy amino acid. We saw the example of serine and threonine, and dicarboxylic amino acid. Other thing that we need to know that when there is L-amino acid present in our body, This enzyme converts it into L-amino acid, and this L-amino acid can convert it into keto acid. Along with that, an ammonia is released. This ammonia again enters the urea cycle. So this is how L-amino acid in our body is broken down to give ammonia. The FMN is used, the coenzyme FMN is used here. During usage of FMN, again regain of FMN is use of O2, which gives peroxide, right? And peroxide is broken down in our body with the help of catalase to water and half of O2. So next is D-amino acid. If it enters in our body, what happens? Basically, D-amino acid is present in plants. It isn't present in human bodies. It is also present in case of microorganisms. And this amino acid which is there, it is said to convert itself into L-amino acid with the help of D-amino acid oxidase. And again the process continues. How L-amino acid broke down, that way it will break down in our body to ammonia. So these two were quite important that we saw. Now if we look ahead, we look at the ways through which we can form ammonia. Okay. First is the ammonia is formed with the help of trans-deamination. We saw the process where trans-deamination, oxidative deamination, non-oxidative deamination, both the process gave. Second is the amino group of purine and pyrimidine also give us ammonia. And biogenic amines are also the sources of ammonia. So these three are the sources of ammonia. Now this next point that we are going to discuss is the transport of ammonia. Transport of ammonia takes place in different ways. The main form in which ammonia is transferred in our body is said to be glutamine. See, transamination occurs in all cells, but glutamic acid or glutamate which is formed has to reach the liver itself. So glutamate dehydrogenase which is present in the liver itself. So we need to convert glutamic acid into glutamine, which is done by addition of one more ammonia. So think, glutamine has two ammonias within itself, and here we remove water. The enzyme which is used over here is quite important. Also energy is used in this process. To store ammonia, the energy is required, and the enzyme is glutamate synthase. So in this manner, glutamic acid is converted into a storage form of ammonia, that is glutamine, which is then taken to the liver and then broken down. Now what happens? Final disposal. That's the same thing that I told you just now. Glutamine will enter the liver and glutamine will break down to form glutamic acid. Now this process which is there occurs by release of ammonia and addition of water. The reverse of the initial reaction, where ammonia is added and water is released. Here ammonia is released and water is added. The enzyme is glutaminase, which breaks glutamine. And the glutamic acid is formed, again undergoes the same reactions as above in the liver, that is trans-deamination or oxidative deamination, to give ammonia, which enters the urea cycle. This was the entire process in which glutamic acid is transported in the form of glutamine. Now we'll see three functions of ammonia. The first function is it is used for synthesis of non-essential amino acids. It is also used for synthesis of purines and pyrimidines. Okay, not all the nitrogens in them, but certain nitrogens are present. Then NH4+, which is formed from ammonia, is used to maintain the acid-base balance in our body.